This presentation was originally given at the ISBioTech meeting in Rosslyn, Virginia on April 11th, 2013. We presented our data that we've generated for the production of soluble recombinant proteins in BACMAM transduced CHO-S cells. This is a continuation of work that we reported on a year earlier where we had used BACMAM transduced HEK293 to produce recombinant immunoglobulin. At that time, we had tried just one experiment with CHO-S cells and had some very interesting results. So we expanded those experiments over the last year to develop a robust expression platform for the production of proteins using CHO-S. So in this presentation, we'd like to present a short overview of BACMAM and what exactly BACMAM is. Talk a little bit about BACMAM in the news, which is basically the literature. And then we have two case studies. First case study is a secreted enzyme. The second case study is a murine IgG. Both were produced in CHO-S cells. And then we'd like to talk a little bit about the conclusions from the studies and also future direction for our work. So BACMAMs were originally described in the middle 90s. They were developed in the lab of Dr. Rick Boyce at Mass General Hospital in Boston. And his original application was for gene therapy. The knowledge that baculoviruses could actually enter mammalian cells has been known for some time. However, the insect cell promoters are known not to be active in mammalian cells. Therefore, none of the genes for the baculovirus are transcribed, and also no proteins are produced from a entry of a baculovirus into a mammalian cell. So what Dr. Boyce did was to replace the polyhedron promoter, which is the common promoter for the expression of recombinant proteins in the baculovirus expression system, with a mammalian promoter. A variety of mammalian promoters could be used, CMV and others. And the current BACMAM viruses carry CMV promoters. What they discovered is that by replacing those promoters, you could now use a baculovirus particle to transduce a mammalian cell and actually produce a gene of interest under the control of that CMV promoter. It was further found that a wide variety of cell types were permissive for BACMAM transduction, and this has been enhanced by the introduction of the vesicular stomatitis virus G protein or fusion protein into the second version of BACMAM viruses, which are illustrated on the next slide. As you can see, in panel A and panel B, we're looking at just a regular baculovirus particle. Could also be a version 1 BACMAM particle because there's no morphologic differences between those viruses. The vesicular stomatitis virus G protein gives the BACMAM version 2 a distinct morphology. And you can see in panels C through F that they maintain a morphology similar to a rhabdovirus, which is what the vesicular stomatitis virus family is. On panel F, you can actually see a baculovirus inside that VSVG coat. And so the advantage of doing this is twofold. First, it has lowered the MOI, or the multiplicity of infection, the ratio of virus to cell that's needed to transduce efficiently mammalian cells by a factor of 10 to 100-fold. So we're now getting reasonable transductions and protein expressions using MOIs below 10. The other advantage is that it did expand the number of permissive cell lines, types of permissive cell lines that could be transduced by BACMAM to include a wide variety of primary cells, stem cells, and even, I believe, a few T cell lineages as well. So turning to the literature, we find that BACMAM is actually growing. Number of reports in the literature are growing. The vast majority of the papers have to deal with either cell-based assays, gene delivery, and then also vaccine production. So in the cell-based assays, this was one of the original applications of BACMAM that has been developed as a useful tool, developed mainly at GlaxoSmithKline for the production of cell lines for use in cell-based high-throughput screening assays. The gene delivery, gene therapy is one that's actually growing more recently. There were at this conference, the ISBioTech conference, some very interesting data presented by groups in Singapore using BACMAMs to deliver genes into stem cells and then using those stem cells to do tissue regeneration, tissue engineering. On the vaccine side, most of the papers deal with the production of VLPs or with the use of the BACMAM particle to transduce cells in vivo so that they actually produce antigen in the animal and then result in immunity to a disease state. There's very few articles on the use of BACMAM in stem cells. But the use of BACMAM for the production of reagent protein, large-scale production or small-scale also, is growing, and there seem to be about 13 papers that we found dealing with that subject. So I'd like to move into the first case study. The first case study is a secreted enzyme. What we observed was different expression levels in HEK293 versus CHO-S cells. This is a 50 kilodalton secreted protein. It's an enzyme, as I said. It's a glycosylated enzyme. It's expressed in the baculovirus infected insect cells at a fairly low level. It's secreted at a level of about 2 milligrams per liter. There are large yield losses and purification, so that system really didn't produce a sufficient quantity of that material, of this protein, to make it economically viable for this customer to use that system. So we were given a starting material of the gene of interest for this secreted enzyme in a gateway entry vector. And then we did some initial expressions in shake flasks. And then our plan is to move into bioreactor with it. We haven't moved there yet. But I'd like to show you some of the data that we've produced in shake flasks. We did our initial screen with HEK293 and CHO-S cells. So for BACMAM transduction, as I said, the cell lines are CHO-S and HEK293. Mediums that we use for CHO is CD-CHO. For the HEK293, we use freestyle medium. We do not supplement the medias. For agitation, it's about 100 RPM. The atmosphere is 95% air, 5% CO2. We keep them at 37 degrees constant. And the MOI for BACMAM for this experiment was an MOI of 6, so that's pretty low. We do find that sodium butyrate, the histone deacetylase inhibitor, does boost expression, especially in the CHO-S cells. So the flasks on the slide here are the shake flasks that we use typically for 1 liter cultures of insect cells, CHO cells, and also HEK293. So I mentioned that the 10 millimolar butyrate was important for expression, especially in the CHO cell line, and this illustrates that point. What we're looking at here is the production of a recombinant immunoglobulin, and you can see that by adding 10 millimolar butyrate, we basically turn on expression. With the absence of butyrate, we really don't see much expression at all. In the presence of butyrate at 96 hours, we were up to 27 milligrams of of recombinant IgG purified per liter of culture. What's interesting is you can also pick up that histone deacetylase activity, or the activity of butyrate, because you can see that the butyrate is freezing the cells into their growth cycle. So the growth cycle is frozen, cells do not divide, and that's exactly what we saw. We see with the butyrate no increase in cell concentration. Without butyrate, the cells grew all the way to 9 times 10 to the 6, but if you remember on the slide before, we only saw expression in the culture with the butyrate. So growth has been uncoupled, and the protein expression has been turned on. So back to the secreted enzyme that we're looking at, we looked at 4 different clones here, and in each case we're picking up expression. This is a Western blot with an antibody specific for this enzyme, and what we're looking at is just expression across 4 different clones at 2 different time points. Really don't see much difference between them, but we do see the protein at about 50 kilodalton, and it does appear to be glycosylated. What was interesting was when we looked at the effect of the cell line on expression, and in this case we're looking at an SDS page, you can see that we have expression very nicely in the lanes 2, 3, and 6, and 7, and those are the HEK 293 cell lines. We really don't pick up expression to any particular degree in the CHO lines at all, even in the presence of the 10 millimolar butyrate. So we don't really understand exactly what's going on with this at this point, but we were interested to see if the protein perhaps was sequestered inside the cell in the Golgi. Maybe it was insoluble or somehow wasn't processed. So we looked inside the cells, soluble cell extracts of the CHO-S cells, and what we're looking here is in lane 2 and 4, the soluble cell extracts, so the cytoplasmic, and the lanes 3 and 5, the insoluble cell extract at a 10x concentration. If you look around the 50 kilodalton marker, we really do not see a large quantity of protein that could be the enzyme in those fractions. So it doesn't appear that there's a large quantity of that enzyme being produced and sequestered inside the cell. So in conclusion, for that case study, the expression profile in HEK 293 and in CHO-S differ. We transduced with the same baculovirus, or the same Bachmann baculovirus. Expression in HEK 293 was as expected. We expected it to be secreted. We were hoping for a high level, and we saw it. The CHO-S cells failed to express at a high level even in the presence of butyrate, and the protein does not appear to be overexpressed in the CHO-S. We're still investigating the cause of this. It could be the sequence, or it could also be possible degradation of the protein. So the second case study is a murine IgG, recombinant IgG that we've been working with for a period of time. We knew going in that this recombinant IgG had a low level of expression. We just didn't realize exactly how low it was. It's a recombinant murine IgG1 and expressed at very low concentration in PEI transient transfections. The specifications for this protein were that it had biological activity in a cell-based assay, and that the endotoxin level was low, less than about 0.5 EU per milligram. Our starting material in this case was a sequence of variable regions, and we designed complete heavy chain and light chain sequences to use with the Bachmann system, and we inserted human signal sequence and codon optimization into these sequences. We had, for the Bachmann generation, we had these genes synthesized into entry vectors for the gateway system. So originally we were working with plasmids that were given to us, expression plasmids that contained CMV promoter and just regular expression plasmids that we used PEI, 25 kilodalton linear PEI, to transfect these HEK293 cells, a co-transfection of heavy chain plasmid and light chain plasmid. And what we're looking at here is an SDS page, and you can see very, very lightly expression in lanes 3 and 4, and again in lanes 7 and 8, but it's very, very low. We know it's expressed because if we do purify, we get the band in lane 10, which is the protein G eluate pool from this expression. But the purified yield on the PEI transient transfections in HEK293s with these original constructs was about 70 micrograms per liter, or 0.07 milligrams per liter. So a very low yield indeed. However, this was active material and it was usable for the cell-based assays, but we were having to do 10-liter productions to get less than a milligram of material. So it was problematic. One of the things that we did when we were screening these was to change our transfection into CHO-S cells to try a different cell line, and we had a tenfold increase in the amount of recombinant immunoglobulin that was produced. So in this case, which was the opposite of what we saw in the last case study, CHO-S with PEI-based transient transfection of the co-transfection of the plasmids gave us about 0.7 milligrams per liter, whereas the HEK cells with PEI gave us 0.07. So we were able to pick up about a tenfold increase. Biological activity in the cell-based assay was the same or equivalent for the HEK produced material and the CHO-S produced material. So that helped quite a bit. But what we wanted to do is we wanted to give a comparison then of what the Bachmann vector could do in terms of expression increase with this murine IgG1. So we were able to do a little bit of a side-by-side comparison. It's not a very level playing field. In the case of the Bachmann, like I said earlier, we synthesized these genes into cloning vectors to go into the Bachmann system. And in the process of doing that gene synthesis, we had the gene of interest, or the heavy chain and the light chain genes, codon optimized for CHO, which I do not believe was done for the original standard expression vectors. The other change that we made was probably even more significant. We included a human signal sequence in those two Bachmann vectors, both on the heavy chain and the light chain, that we knew gave us good results with other recombinant immunoglobulins. So we ran 10-liter bioreactor cultures of both of these. On the PEI side, we ran a 10-liter bioreactor with a standard co-transfection using 25 kilodalton linear PEI. Both of those vectors had the CMV promoter. They have a murine signal sequence. They were not optimized for CHO. The Bachmann vectors, as I said earlier, have CMV promoter as well. Two Bachmann viruses were created, one for heavy chain, one for light chain, so we did a co-transduction. And they do have a human signal sequence and also were codon optimized for CHO. So the bioreactor parameters were very similar. The cell line was CHO-S, same cell line. The media was the same, so we used CD-CHO from Life Technologies Corporation in both cases. Neither of those 10-liter bioreactors were supplemented. Our pH was maintained between 7 and 7.4. The DO levels were the same, 40% of oxygen and air. We kept the temperature constant at 37. We put in a standard amount of plasmid for a 10-liter bioreactor, which was 10 milligrams of plasmid. So we put in 5 milligrams each of heavy chain and light chain. And we used 25 kilodalton linear PEI, a total of 40 milligrams for the 10-liter culture. On the Bachmann side, we had a cell line, the same cell line, Same parameters. For BACMAM, the MOI was 18 total, which was a little higher than what we had done for the secreted enzyme that we were talking about earlier. But our experience with recombinant immunoglobulin has been that if we could keep the MOI somewhere around 20, that seems to give us fairly good results. So we used an MOI of 9 for each for the heavy chain and light chain BACMAMs. No PEI was used, and in both cases, the cultures were given 10 millimolar sodium butyrate at either the point of co-transfection in the case of PEI, or in the case of BACMAM, the co-transduction. So a point on the MOI with the BACMAM, we found that we needed to concentrate the virus in order to get the MOI that we wanted without having to add too much of the virus stock to the culture. We try to stay below 10% of the culture volume as virus stock, because these BACMAMs are made in insect media, and we're trying not to just perturb the culture by adding large quantities of insect media into the mammalian bioreactor. What we used was a system that we've used for quite some time. Protocols available on our website, and it's a concentration method based around hollow fiber. This is a 500 kilodalton molecular weight cutoff hollow fiber filter from Spectrum Laboratories. Very simple system. We just use a recirculating pump, a peristaltic pump, and these can be sized for the size of high titer stock that you want to process. We process non-aseptically. It's not a sterile flow path. We filter sterilize the virus once we're finished concentrating. And you can size the filter so that you can get your process done, say, within an hour or maybe 2 hours. And we process this right before we put it into the bioreactor. So the efficiency of the hollow fiber has been demonstrated in previous talks that we've given, and this slide illustrates that, where we find, if we're looking at plaque titer, and this was actually done by plaque assay, we found our original titer in the load was about 2 times 10 to the 7th. In the permeate, we found no colonies or no plaques for the baculovirus, all the way down to undiluted permeate. And then in the concentrate, we're up to over 1 times 10 to the 8th for the plaque forming units. So, and this that correlates with the amount or the degree of concentration that we did with the virus. So back to the 10-liter bioreactors, then we put in the virus at an MOI of 18 total and a standard PEI transfection. And what we found with some of the bioreactor parameters, the viable cell count in the BACMAM transduced bioreactor stayed fairly constant over the 72 hours that we processed, or that we allowed this to go post co-transduction. So it drifted from about 1.1 times 10 to the 6th up to about 1.4 times 10 to the 6th. The PEI, we saw a drop-off in the viable cell count after 48 hours. So between 48 and 72 hours, we had a drop in culture viability, therefore a drop in viable cell count from about 1.4 times 10 to the 6th down to about 6 times 10 to the 5th. And this is also illustrated on this slide, which is just the percent viability for the bioreactors. You can see that the BACMAM co-transduced bioreactor maintained viability fairly nicely, whereas the PEI bioreactor, we had a drop in viability below 40% at 72 hours. And this is somewhat typical of what we see in a PEI-based transfection of CHO-S under these conditions. Purification scheme was the same for both of these expressions, and is a little bit different. So I thought I would put up the purification scheme that we used. This antibody basically does not like to be processed in filters. So that makes it problematic for concentration. We've lost a number of times lost material when we tried to concentrate through a Amicon Centratec uh, uh, filter, ultrafilter. So what we did was we used MEP hypercell to grab the immunoglobulin out of the culture medium. With MEP hypercell, we don't need to dilute the culture medium, so we could just process the 10 liters. We use 10 grams of MEP per liter of culture, and we batch bind for about 2 to 4 hours. We add 1 millimolar EDTA to that to bind up any potential iron chelators that are used to present iron to the cells that may interfere with the binding. We then take that, pack it on a column. We isolate the resin, pack it on a column, wash it, and elute at pH 4, neutralizing right away to pH 7 with 1 molar tris. We then take the MEP eluate directly onto protein G sulfurose fast flow, wash with PBS, and elute with typical conditions, 0.1 normal glycine, pH 3, neutralizing to pH 7 with 1 molar tris pH 9 right away. We also add 10x PBS to make sure that there's salt present in those elution fractions. So at that point, what we would normally do with an antibody would be just to take it onto an ultrafilter, probably a 30 kilodalton or 50 kilodalton molecular weight cutoff filter, and concentrated either in the centrifuge or in a stirred cell. But we know with this antibody that it binds to surfaces very readily. So what we did was used 1 ml columns of MEP to concentrate, to capture back the antibody that we had eluted from the protein G. And then we took that directly into dialysis against PBS. We sterile filtered it and we vialed it. We found that this scenario was the best one for limiting the loss of this antibody in purification. So this shows the MEP step. On lane 2, we have the MEP load. Lane 3 is the flow through, and as you can see, the MEP pretty much grabs everything out of the culture supernatant. We then wash, and then we have our elution fractions, and you can see the IgG band. Lane 11 is the final protein G eluate pool prior to concentration and dialysis. So this is the material that was produced in the 10-liter bioreactor for the PEI cotransfection of the CHO-S cells. So turning to the CHO-S BACMAM 10-liter, this is a similar type of illustration for the MEP step. We had the MEP load, flow through. Again, it was pretty much capturing everything. We have the MEP elution pool, and we had actually 2 pools in this case. And then we had the protein G elution pool from the protein G step. Yield comparisons were interesting. Remember, originally we started out with HEK 293 cells with PEI cotransfection. We had a yield of about 70 micrograms per liter, 0.07 milligrams per liter. If we switched into the CHO-S cells, we saw 0.7 milligrams per liter purified, and that held up when we ran the 10-liter bioreactor as well. We saw a 0.7 milligrams per liter yield, purified yield with PEI-based cotransfection. The cotransduction of the CHO-S cells with the BACMAM gave us a 5-fold increase in the yield to 3.5 milligrams IgG purified per liter of culture. So that was very nice. So in conclusion, the expression profiles for this antibody and for also the secreted enzyme differed in HEK 293 and CHO-S. And what that's taught us is that when we're looking at new proteins, we should screen our cell lines and look for a cell line that is optimal for the protein that we're expressing using BACMAM. BACMAM constructs, they scaled to 10 liters just fine. But right now we're still at low cell density. We're transducing at 1 to 1.5 times 10 to the 6 cells per mil. Uh, and we're getting good results, which is due to the virus concentration, but we still think that we should be able to transduce at least 2 to 3 times 10 to the 6 cells per ml. I think a key there is our ability to take and concentrate the virus down, possibly diafilter it into a buffer that's more compatible with the mammalian cells in the bioreactor. So we also know that the proteins produced using BACMAM retained biological activity, both with the enzyme that was active and also the recombinant immunoglobulin that was active in the cell-based assay. And we know that the activity is equivalent to proteins used or proteins produced using PEI. So our future direction is basically to try to increase the cell density at transduction and to expand into the production of other proteins, including virus-like particles, which we're currently involved with doing in HEK293 cells using PEI transfections. So we're very interested in switching some of those genes over into the BACMAM system and applying that to the production of VLPs. So basically, I'd like to thank the people that was working on the project, the ISBioTech organizers for allowing us to present our data at the ISBioTech meeting. Also like to acknowledge April, Anita, Pat, and Kay Lee from Kempbio for their contributions to the work, and also our friends at Medigen, Peter, Irina, and Rakhmat for their molecular biology expertise. Thank you very much for listening, and I appreciate it.